And let's share my screen. All right. So I'm gonna close off my video so to save some Wi-Fi resources. All right, let's get started. All right, so on Tuesday, we talked about how we can use the data. So first, we can use the data to calculate the genetic distance between A and B and C, right? So we can get the estimation for the recombination frequency, RAB or RAC or RBC. And once we have this numbers estimation for the recombination frequencies, we can put the three loci A, B, C into the right order based on some criteria here. For example, we can use SAR, which is the sum of adjacent recombination coefficients. So always the right order will give you the smallest number value of the SAR, right? Okay, so this is how we can prove this. So as I mentioned, this is very important. I hope everyone understands this because it is very likely for me, the probability is very high for me to put a question like this on your final exam. So make sure you understand how we prove that the right order gives you the smallest number of the SAR value. All right. So alternatively, we can also use another measure, the additive measure, which is X, to represent the average number of the crossing over between these two loci um, per meiosis. So this is another thing we can construct. Sad. Okay, so the sum of the adjacent distances. So still, the right order will give you the smallest number of this thing, and also we can use the likelihood value as well. So this will also can help us to find the right order of these three loci. Okay, so let's look at an example here. Suppose we want to extend our problem into from three loci problem to a five loci problem. So here, suppose we want to find the right order for five loci, A, B, C, D, E along the same chromosome. All right. So this is still a very small problem. So because we're only dealing with five loci, so totally we only have like 60 possible orders for these five letters. So the five loci. So if we start from two loci A, B, all right, and okay, that's easy. We can use the data. So either the backcross design progeny or the F2 progeny, right? So we can use either experiment to generate lots of progeny. So, and by looking into the progeny, we can use the data to calculate the recombination frequency between A and B. So that's the first step. And then next we want to put C in. So there are three possible ways to put the C in this linked group, right? Either C will be the first one, or C either A will be in the middle, or C in the middle, or B in the middle. So three possible ways to put these three letters. So then we can use what we have learned, right? We can use the SAR value to calculate the values for these three possible orders, and the one with the smallest SAR value will give you the right order. Okay, And after we're done with this, we can continue adding another letter loci locus into this into this chromosome, right? So for example, from here, oops, let's do this, sorry. So suppose this is the right order. So if we want to put another letter in, say D, so there are four possible ways to put D in, right? All right, and we just extend, we just repeat the calculation of the SAR value for these four different orders to find the best one, and we continue to do this, and we can calculate the SAR values for all 60 possible maps. All right, that's not too difficult because we only have like 60, right? So if we can examine all 60 orders and calculate the SAR value for all these 60 orders, this is called the exhaustive search. Since we are only dealing with like 5 loci, as I mentioned, so the cost is not that high. So it's low. So we can afford a exhaustive search. However, any questions here? Any questions?
Just one clarification question, sir.
Yeah, sure, right. Yeah.
The numbers that come after the combinations is in fact the SAR value that you have described.
Let me see.
Or is that
you mean this question? You mean these numbers, right?
Or is that a percentage?
Well, you mean these numbers?
Yes, sir.
Let me see. So I didn't pay too much attention about this. Yes, I believe so. So because I just took this example from the textbook, I didn't look into the detail, but I believe you are right. Okay, so this should be, because there's no, so if we are talking about 2 loci AB, so there's no, there's only one recombination frequency, so we don't have to calculate the SAR, right? So that's R, I guess, RAB.
But then we can make an assumption that BA, the BA combination would be might be a higher value, and therefore this is the least possible. Most probably.
You mean the BA, right? AB is equal to BA because so in this case, we do not consider the direction. We only consider the order. So you can either from left to right, or you can go from right to left. So they give you the right. They should give you the same answer. Yeah.
Okay.
Yeah. Because yeah, right. So I need to make sure here the order means the relative order, not the direction. So if we want to get the exactly order, so A, B, C, D, E actually is different from E, D, C, B, A, right? But in our case, these two are the same, identical.
I understand. Thank you.
Okay. Yeah. So I think you are right. So this value, besides each map, gives you the calculated SAR value. So for example, here, I believe this is the right order. Oh no, smallest one, sorry. Jeez, this is a huge mistake. Okay, this will gives you the right order because the smallest one, right?
So I have a question.
Yes.
So the exhaustive search is searching all the combinations, but if I first determine the ABC's order, then I just follow one like sub-line. So it's not the exhaustive search, right?
That's right, yeah. So here I just give you an example, but you can develop a very smart program. So at this step, if you already determine this is the best thing, so you can ignore this path at all, right? You don't want to consider this at all. So you can just go along this trace to find to find the right order in a much easier way. Yeah, but here I just want to show you the concept, the definition of the exhaustive search, because in this case it's not that complicated. We only have like 60, so you can do all of them, right? And find the best one. But yes, so once you are considering lots of loci, which is the case usually we deal with in the reality. So you need to find a better way, a smart way to help you to find the solution quickly and inexpensively. Any other questions? Okay, so this is the exhaustive search. All right, now, as I mentioned, so it's okay if you use the exhaustive search when you are dealing with a few loci. So, but if you increase the number of the loci, what you're gonna see is like the calculation cost, the computational cost will increase sharply, right? So if you are dealing with like 5 loci, you only need to evaluate 60 orders, 60 maps, different maps. So you only need to calculate the SAR values for 60, and you can find the best one, the smallest one. However, if you increase just one, so if you are dealing with like 6 loci, so you have to like deal with like 6 times more calculation, 5 times more calculation, right? So if you keep increasing the number of the loci in your problem, so eventually you cannot afford that. So this is the limitation for the exhaustive search. So the exhaustive search algorithm is usually used to deal with the problem one, the number of loci is less than 10. Any questions? Okay. So yeah. Let's go back to Jinli's the question. So yeah, of course we have some better way to deal with this. So other than the exhaustive search, we have the heuristic search, right? And which means like we at the beginning we just randomly assign a initial order, and starting from that initial order, we can continue. We can do some iterative search to help us to find not the best one, the one close to the best one usually. All right. So there are 2 possible ways to do this kind of search. One is the complete global rearrangement. Suppose we have A, B, C, D, E. Suppose this is the initial map, initial order. So, so for this one, we can calculate a SAR value. Okay, and so this is the first step. The second step, we can randomly randomly reorder these five letters. For example, we can put this into this order, randomly shuffle these 5 letters, right? We can, so this is step 1, this is step 2, and we can calculate the SAR number 2, number 1, right? Okay, now once you're done with the second step, you just compare these 2. So which one is smaller? So if the first one is smaller, just discard the second one, and from here you can run. So you just, if the first one is the smallest one, you just ignore this one and just remove this one and keep the first one, and do the same thing. Do the same thing. The step 3, you do the same thing, reshuffle these 5 letters to generate another random order and calculate another SAR value to compare to the one which is left. And you continue in doing this until you find the best one within a number of iterations. For example, you can run this program for 100,000 times. So although you cannot evaluate all possible. So suppose we're dealing with not just 5 loci, suppose we're dealing with like maybe 20 loci. So if you want to do the exhaustive search, so that means like you have to deal with, I don't know what's the number, but probably a very big number because we cannot afford that. So you just do this heuristic search, and then so you only need to like do the iteration for probably 100,000 times, and then among those limited search, you can find the best solution. Is that clear? Is that clear?
Could you go over that again?
I'm sorry?
Could you go over that again, please?
Yes, of course. Okay, so let me put this. Okay, so suppose we have 5 letters, right? A, B, C, D, E. And because we don't know the right order, so we want to evaluate, we want to search the entire base, or maybe subspace to help us to find either the best map order or the optimal map order, which is very close to the best one. Suppose we are dealing with like this 5, so we give the initial order of this. So based on this order, we can calculate a SAR value. Is that correct?
Mm-hmm.
Okay, this is the first step. Now let's look at the second step. What is the second step? Second step is like, we just randomly shuffle these 5 letters. For example, you can end up with this order, right? Map. Am I right?
Mm-hmm.
And this is the second step. So yeah, based on this randomly shuffled order, you can calculate another SAR value. Let's say prime, okay? Now you can compare this two. So which one you want to, so suppose you have 2 SAR values here. So which one do you want to keep? The smaller one, right? The smaller one.
Yes, the smaller one.
So yeah, of course. So the smaller ones, for example, if this one is smaller, which means the initial order is better than the second order, right? Do you agree?
Yep.
Okay, then we can discard this one. No, we don't worry about this. We just discard this one, right? We keep the first one. Now we do this. We do the same thing. The third time, we can reshuffle this again. So probably it will give you another order. Oops, A, C, D, B. Okay. So this is the step 3. Also, based on this new order, you can calculate another SAR, right? Now you compare this one with the one with the one left from the previous step. Do you agree?
Mm-hmm.
So you can do the same thing. So which one is better? Maybe this time this one is better because it's smaller. Then you just discard this one and keep this one as the current. Is that okay?
Yes.
Yeah, you continue this. You do the 4th, 5th, and so on. All right. So imagine, so we are here, I just, for simplicity, I just put 5 letters. Suppose you are dealing with 20 letters, 20 loci. So if you do the exhaustive search, so this will probably gives you this many. Oops. Jeez. This many possible orders, let's say. Is that true? Many. So if you are dealing with like 20 loci, so probably if you want to do the exhaustive search, there are too many orders you have to evaluate. So that's impossible. That we cannot afford that. But what we can do is to do this. Instead of doing the exhaustive search, we can do this heuristic search. So when we do this kind of iterative search, we only go that far. So we only need to do this many times. Although we didn't search all the space, all the possible maps, but still good, because this strategy will give you something we call the local optimum, which is very close to the global optimum. So we want to identify the ideal situation is like, based on the data, we want to find the global optimum, which is the real, true, right order, which is the real, true, correct order for this 20 loci. Is that clear? Is that clear? But if we do this kind of heuristic search, we cannot search the whole base. What we can get is we can find the local optimum, which is not the global optimum, but still it is very good because it is close to the global optimum. Am I clear? Yeah, you are lucky. If you are lucky, you can, even within this limited search, you can find the global optimum. Yeah, if you are lucky, there's a chance. But usually, like this kind of algorithm only gives you the local optimum rather than the global. So this is the limitation. Is that clear? All right. Okay, I just talk about the complete reordering or rearrangement, because when we talk about the 5 letters, each time we totally reshuffle these 5 letters, right? But we can also do a even less expensive way. So we do the shuffling or the rearrangement for partial of the letters, not the 5 letters. So for example, if we start from this A, B, C, D, E, next time we can only shuffle C, D, E, the 3. So for example, if the first order is, initial order is A, B, C, D, E, right? Next time we can evaluate A, B, D, E, C, because we only shuffled the 3 letters. That's the partial rearrangement. So different way. So people, because there's a trade-off. Because the best way is to do the exhaustive, right, search, because that can guarantee you find the best map, the global optimum. But because when you deal with a very complicated problem where lots of loci are involved, so you cannot afford that. So you need to find a better way to balance the computational cost and the accuracy, right? So there's a trade-off. So this is not a bad idea. So usually, like, yeah. So any questions? All right. So this is so-called the heuristic search, and there are other 2 types of heuristic search. I don't want to spend time on this, so I will leave this to yourself. Please read the textbook. Those 2 methods are not required, but for your own benefit, please read the textbook and make sure you understand what we do here. All right. Okay, so yeah, I just mentioned this graph just now, right? So now you understand this, is that right? Because we have a whole search space. So suppose we're dealing with like 5 letters, 5 loci. So we need to evaluate. So the exhaustive search means we need to evaluate all 60 possible maps. So if you do that, it will guarantee that you will find this, the global optimum, the best map, right? But if we are dealing with too many loci, so we cannot do that. So we do the non-exhaustive way, but this way usually can help us to find the local optimum, which is very close to the global, but probably it's not the best one, but still good. So this is how we do this kind of search using different algorithms to help us to use the data to find the right order of this loci on the same chromosome. Is that clear?
Yeah.
Is that clear? Any questions?
I have a question. So if it's randomly rearrange the orders, why it depends on the initial order? So because the first is randomly select, and second one is also random select. Why the initial order is important?
Yeah, good question. So actually this depend on the initial order probably will not affect the complete rearrangement too much, but it may affect the partial rearrangement. So yeah, that's a good point. So if you do the complete rearrangement, probably it doesn't matter which order you chose for the initial value, right? Initial order. But if you want to do an even less expensive way to do the search, for example, if you do the partial rearrangement, then probably the way you initialize your map order probably will affect the final results. Yeah, does that make sense? Okay, very good. Any other questions?
So would that initial order be affected by the flaws or the type of sequencing done? Would it be affected by previous information from other genes and model?
So yeah, that's a good question. So I think the question was very good, but I think I need to answer your question in this way. So here we only talk about the computational skills. Suppose we do not know any prior information, biological information. So for the initial value, we totally random guess where these 5 loci are located, right? So in this way, if you put the 5 letters in a totally arbitrary way, so this will affect the results, final results, if you use the partial rearrangement way to do the search, right?
Got it.
However, we can do better because I think go back to your question. So if you are a combination of the statistician and the biologist, so you can efficiently use your sequence data to get some prior knowledge. So you can give a better initial value. You can give a better initial order for this 5 loci. So that means like you are one step forward to the goal than other people who do not have any prior knowledge about this. Does that make sense?
Yep.
Okay. Yeah. So so that's very good. So here we're just talking about the pure computational skills. Suppose we do not use any prior information, so then we can only randomly put these 5 letters in random order. So there's no benefit of doing this, right? But if you can leverage any information, biological information, sequence data to help you to get a better initial value, initial order, then bingo, you're closer to the goal. Yeah. All right. Any other questions? Good. Okay, let's do this. Okay, so since in most of the cases we do not do the exhaustive search, we use some smarter but smarter way to do this. But this method may not give us the best solution or the global optimum. So usually when we get a results from this kind of from any of the heuristic search, we need to check the results. How confident the map we generated. So how confident we are about the map we generated from any of this heuristic search. Is that correct? So we need to we need to construct some confidence for the calculated or derived map based on any of this heuristic search method. All right. So before we talk about the confidence interval, the confidence of the map. So let me introduce some very important, basic but important concepts in statistics to you. So first, what is statistics? What is statistic? Can anyone tell me? What is so you all have taken statistics class, so you should know, you should have known the definition of statistic, right? What is statistic?
Using the existing data to infer the parameter in the population?
Yes, yes, yes. Okay, so statistics, so the whole discipline of statistics is to do one thing, actually. So yeah, although there are a lot of branches, but the entire statistical society are doing one practice is like, we want to study, we want to make some inference or conclusion about the population we are interested in, right? So suppose here we have a population of interest. We want to study the population, and we want to know, for example, for simplicity, we want to know the average of the population. However, the population is too large to study, so you cannot. For example, if we want to study the average height of the human beings on this planet, you cannot survey each individual, right? Because that's impossible. That's too expensive. So what can we do? So to study this population, we can take a random sample from this population. If the random sample is representative of this population, then we can study this sample and get some conclusion from this sample and go back to make the inference for this population. Is that clear? That's the statistics. Okay, all right. So the statistic practice is actually here. So the target is the population. We want to study the population, but the population is too large. We cannot study the whole population. What we can do is we get a random sample from this population here. And if the sample is a random sample, which means this sample is representative of the population, then we can only study this sample. That's easy, right? We can calculate the sample mean. Because you cannot calculate the population mean because you cannot get the data for the whole population, but you can easily get a sample and calculate the sample mean and use that sample mean to go back and infer the population mean. All right, now the sample mean is called the statistic. Now this is the key point. So anything you concluded from the sample is called the statistic. And you use that statistic as an inference for the population. So in this case, we calculate the sample mean, and the sample mean is the statistic, and we use that as an inference for the population mean. Is that clear? Does that make sense? Okay. All right. So, okay, now sampling distribution. This is another important concept. So, okay, so suppose this is the whole population. Okay, maybe I can randomly sample. Maybe I can sample a random sample of size n, and from my sample, I can calculate a sample mean, right? That's the statistic from my sample, right? Yeah, maybe Jinli, you can sample another sample of the same size n, and then from your sample, you can calculate your sample statistic. And so that's Ryan, right? Ryan, he can sample another random sample of size n, and he can also calculate the sample statistic from his sample, right? Right? Now, do you believe all these statistics are identical? No, because of the sampling error, right? So if you plot, so suppose we can generate lots of this kind of random sample, and from each sample we can calculate a sample statistic. And if you plot these statistics also follow a distribution. That's the sampling distribution, right? And the sampling distribution actually, the center of the sampling distribution should be a very good estimation of the parameter, the population parameter, which is the population mean. All right, we try to use the sample statistic to infer the population parameter and the statistic. The parameter is the thing we want to estimate, infer, and the statistic is the thing we use to infer the parameter. Is that clear? Okay. All right. Now, this is the sampling distribution. The reason why we see the variability is because the sampling error. And this is how we calculate the sampling error, right? Any questions here? All right, so this is a very common situation. So if we can afford multiple samples, but in some cases, we can only afford one sample. So suppose here, only I can afford to get a sample from the population. We only have a single sample here. So how can we get the distribution of the sampling distribution? So there's one way we can do is the bootstrap, okay? Suppose your sample, the single sample is a random sample, then the sample distribution should reflect should be very close to the real population distribution. Do you agree? Okay, now, so if you have your own sample, you can keep sampling your sample to generate multiple samples by replacement. So are you familiar with the sampling by replacement? Okay, good. All right. So for example, you have like a sample of one. So you have a sample of 1, 2, 2, 3, 3, 3, 4, 4, 4, 4. Okay. So this is your sample. And your sample consists of 10 elements, right? Okay, so the bootstrap sampling means what? Means like from your sample of 10, you generate, let's say, generate another sample of 10 from your sample. So because in your sample, which number is the most frequent number? 4, right? Which means you have more 4s in your sample. Then the chance for you to sample 4 again is higher than other numbers, right? Do you agree? If you randomly sample a number from your sample, which number is the one has the highest chance to be resampled? 4, right? Okay. Now, so if you do this by so for example, if you sample just so this is your initial sample. Now you want to generate a bootstrap sample. Let's say bootstrap 1 from your initial sample. Okay, the first number you sample from the bootstrap sample 1 probably is 4 because it's more likely for you to sample a 4 from your sample, right? 4 is most frequent. The second one probably 4 again, and the third one 3. The fourth one 4. The fifth one 2. The sixth one 3. The seventh one 1. The eighth one 4. The ninth one 4. And the 10th one 3. So this one is likely, right? This is the okay, which means like once you sample the 4, you put the 4 back to keep the to keep your initial sample a complete one. Replacement, you put it back, and then you do the resampling again for the second one, right? So this is the replacement sampling. Okay, now you complete sampling the first bootstrap sample. Do you believe this sampling is possible, is reasonable? Okay, now you can keep doing this. You can generate another bootstrap sample. Do you agree? Something like this. Because 4 is the most frequent element in your initial sample, so 4 is more likely than other numbers to be resampled in your bootstrap sampling, right? Okay, now by doing this, you can generate lots of samples from bootstrap. Do you agree? And each time you can calculate the statistic from your bootstrap resample. And you can generate a distribution of your statistic, and then this is the bootstrap distribution. Is that clear? Any questions? Very good. Chat, let's see. Okay, good. All right, thank you. Okay, now so far we have learned the concepts of the statistics and the bootstrap sampling. We're gonna apply this theorem to our problem. So let's go back to our problem here. What is our problem? What is our problem? We want to generate some confidence for the derived map, right? So we just talk about how we can use different search skills to help us to find the right order for these 5 letters, right? Suppose we are not interested in the whole map, but we want to see if within this map, if these 2 loci are adjacent to each other. So this is what we want to test. Okay, so this is the map, optimal map from your search algorithm, non-exhaustive search algorithm. And because we are not interested in the whole map, but we want to see if these 2 letters are really adjacent to each other. This is what we want to test. So we can, because we only have, how many samples do we have? We only have 1 sample, right? We generate 1 experiment, either a backcross experiment or a F2 experiment. So we produced a lot from this cross. We produced a large number of progeny, and based on the progeny genotypes for these 5 loci, we can calculate the R's, the recombination frequencies between any 2 letters out of this 5, and then we can use the search algorithm to help us to construct an optimal map like this, right? And here, because map, this optimal map tells us that the loci D and B are adjacent to each other, because this is of this is of interest to us. So we want to test how likely or how confident these 2 loci are adjacent to each other. So we can use the bootstrap to construct a confidence on this segment. All right, so now we can draw a large number. So here, the big N is the number of bootstrap samples. Anyone has problem with the bootstrapping? Okay, good. So because we have a single, we have a single map population or sample, right? We only have 1 sample here. Now from this a single sample, we can draw a large number of bootstrap samples based on the bootstrapping. And when you get, so for each of this bootstrap sample, you can derive a optimal map. Can we? From each bootstrap sample, can we derive a map for these 5 letters? Do the same thing, right? So just like what we did for the original sample. So from your original sample, so we have like many like progeny from the experiment, right? We use the data to construct a map like here, right? Now you do the resample based on the bootstrapping. you can end up with another bootstrap of the progeny. So from your initial progeny sample, you can bootstrap. You can do the bootstrap to generate another bootstrap sample, which consists of progeny. This progeny are resampled from the initial progeny. Can you follow me? And once you get a bootstrap sample, you can follow the same strategy to construct a map, optimal map for that specific bootstrap sample. Can we? Any questions?
Just repeating what you said. We're finding the optical combination for DB here, and we're doing that for one particular individual. But then we are also saying that we can do bootstrap samplings for the progeny of the individual.
Okay.
As another possible calculation.
Yes. Okay. So let me draw this diagram to help you better understand this. So this is your initial sample. What is the initial sample? The initial sample are the 1,000 progeny you created from your own across experiment. Is that correct? Either backcross or F2.
Mm-hmm.
Okay, now use this. You can generate a map like this. This is what we have learned. Is that correct?
Yes.
Okay. Now we want to see, so this is your results. So without any resampling, this is your results. And from your derived map, you see B and D and B are adjacent to each other. Now you want to test how confident we are about this two letters are adjacent to each other, right?
Mm-hmm.
Okay, now we do the resampling. So from this sample, we can draw another 1,000 progeny. But this 1,000 progeny is very close, very similar to the initial 1,000, but not identical. Because what? Because some individuals from the initial sample may be sampled for more than one time here, right? Is that clear?
Mm-hmm.
So when we do the resampling with replacement, we can generate another bootstrap sample, 1, which consists of 1,000 progeny. This 1,000 progeny are resampled from the initial sample. Is that clear?
Yep.
Okay, now, once you have the first bootstrap sample, so you can do the same thing to generate another map. Maybe the map generated from this bootstrap sample are identical to the first one, which is good, which means, oh my God, will build up the confidence for the initial calculation, right? Do you agree?
Mm-hmm.
Okay, now you do the same thing from the initial sample. You can generate another bootstrap sample. Can we? Yeah, this bootstrap sample 2 should be very similar to the initial sample, or should be very similar to the first bootstrap sample. Do you agree? But they are not identical because of the sampling with replacement.
Mm-hmm.
Okay.
They shouldn't be entirely the same.
Entirely.
Not.
They're very close. They're very close. But yeah. All right. Now with this, can you derive another map? So maybe this time it's different. You end up with this. It could be, right? But still, B and D are adjacent to each other. So this data also support your initial map. Do you agree?
Mhm.
Okay, now you repeat this for 100 times, which means you generate 100 bootstrap samples using the same strategy. Now you can generate 100 maps. Can you?
Mhm.
Now, out of this 100, you can count how many maps carry B and D adjacent to each other. That will give you the confidence. So suppose you did this bootstrap resampling for 100 times, and out of this 100 times, you get 96 maps carrying B and D together. So this will give you the 0.96 confident that your original map is correct in terms of B and D.
Mhm.
Is that okay? Good, very good. Thumb up. Okay, nice. All right, let's keep moving. All right, I'm done with this chapter. Any questions before we move to another topic? So what have we learned in chapter 3? Can anyone help me to summarize? What do we learn from the chapter 3?
Determine the right order of many loci.
Loci, yes. So still, so this chapter 3, should be based on chapter 1 and chapter 2, right? Let's see. Okay, so that's okay. Very good. We also introduced this. What is the statistic bootstrap? A parameter, population, sample, SAR, order of the loci algorithm. Yes, yes. All right. This chapter 3, should be based on chapter 1 and chapter 2, right? Chapter 1 is about like the map function. So we need to worry about the genetic map between A and B, which is the R, recombination frequency. And the second chapter, we talk about how we can calculate, estimate that R, and we can test if that R is equal to 0.5 or significantly less than 0.05, which indicates the linkage between these two loci, right? So that's what we learned. Now, chapter 3, so we extend to the problem of 2 loci to the 3 loci or more. So because we want to use the calculated R value, the recombination frequency, to help us to find the right order of this loci along the chromosome. All right? Do you appreciate the logic behind the chapter 1, chapter 3, and chapter 3? Yes, okay. All right, so chapter 4, we would like to use what we have learned so far to help us to analyze multiple loci, Mendelian loci along the same chromosome. All right, so here, before we move forward, so we need to introduce another important concept in statistics, Markov chain. So Markov chain is a random process. So statistics usually deal with random process. So Markov chain is one of the random process that undergoes transitions from one state to another. So for example, here, Markov chain could be the transition between these two states, states E and states A. And so if the current state is E, so next step, so the current step, if the current state is E, so next step, there are 2 possible states. Either the E will stay in this, so either the process will stay in the same state, will stay in the E state as well, or maybe next step, the whole process will transit from E state to the A state. So then there are 2 probabilities, right? The probability that the process stay in the same state, E, is 0.3. Or the probability that next state is A will be this. So these two probabilities should add up to 1. Is that clear? So suppose the current state is E. So there are two possible outcomes, right? For the next step. The next step, either the process, the whole Markov chain process will stay in the same state, E. The probability of that is only 0.3. But the next step, if the state becomes to A, then the probability is about 0.7. The same thing for A. So once the current state is A, so then for the next step, if the process stay in state A, should be 0.6. But there's another probability that the state will be transit back to state E with probability of 0.4. So this is the Markov chain. And there's a property for Markov chain, which is the probability distribution of the next state only depend on the current state, but not depend on the state before. So if you want to see the process, the next step for the process, you only look into the current state, and the current state will determine the next state. All right, so this is the Markov chain. All right, so Markov chain is a very useful model. In the future, you're gonna see like we can use Markov chain to do a lot of things. We can use Markov chain to model the DNA sequences, because if you look at the DNA sequence, right, actually from here to here, and from here to here, and here, this follows a Markov chain, right? So in this case, we have 4 states here. So, and the specific DNA sequence is actually the transition among these 4 states. So if you start from a T here, so the next sequence, the next letter from the current state could be either T itself, or it could be changed to A or C or G. So this is a Markov chain model, a classical Markov chain model. In our problem, we can use Markov chain to help us to calculate the joint probability of a specific progeny. So let's look at this example here. So we always start from the simplest example. So suppose we are dealing with a backcross design, and we are considering 3 loci here, A, B, and C. And A, B, C represent the 3 steps along this Markov chain. So let's look at the current current state, because this is a backcross design. And so if we look at a single progeny, okay? And at the first locus A, so there are only 2 possible genotypes. Do you agree with me? Because this is a backcross design. So if you look at just A locus, there are only 2 possible genotypes. So these 2 possible genotypes defines the 2 states, actually, the state 1 and state 2. At this step. So, okay, suppose for this progeny, at this locus, the genotype is 1. The initial step is 1. When we look at the B locus, suppose B also carries the genotype 1 here, all right? Which means from step 1 to step 2, there's no state change, because state 1 here and also state 2 here. Can you follow me? So what's the probability of this? So based on our the knowledge we have learned so far. So if for locus 1, for locus A, if the genotype is 1, state 1, and for locus B, the genotype is also 1, B1, B1. So what's the probability for this transition? Stay in the state 1. Can anyone tell me? Let me give you some hint. I'm sorry, say that again.
1 minus R.
Yes, okay. Because there's no crossing over, right? So the probability for this transition from step 1 to step 2, if the entire Markov chain, so the whole chromosome is a Markov chain when we talk about this. So the chain stay in the same state, 1, the probability is this because there's no crossing over. Do you agree? Okay, let's look at the next step. So now if we look at the C, so we forget about the genotype of A because the Markov chain, the next step only depend on the current step, right? So currently we have state 1 at this B locus. Let's look at locus C. So suppose the locus C, this progeny carries this genotype. All right, so there's a transition from state 1 to state 2. So what's the probability here? Do you agree? Okay. So I hope it is clear to everybody. So right now we're talking about a Markov chain. So just think about the whole chromosome is a Markov chain from A, B, C. So this is a chain. And each locus represents a step. And any step, there's only 2 states, either 1 or 2. So this 1 state or 2, state 1 and state 2, actually denote the 2 genotypes from this backcross design, right? Any questions? Okay, good. All right, so to go further, so let's study another very important concepts here. So we're gonna use this very often in the future in this class. Okay, the conditional probability. So what is the conditional probability? The conditional probability is actually based on the part of the sample space, all right? So there are 2 things, right? The absolute probability and or the conditional probability. So when we talk about, let's look at this first. So when we talk about the absolute probability, let's say probability of A, which means the probability of the event A. So when we talk about the absolute probability, this is referring to the entire sample space. So here is the entire sample space. And the probability of event A is actually represent this area in relative to the entire square sample space. Is that correct? So if we want to calculate the probability of A, what is that? It's the area of circle A, right? Divided by what? The whole sample space. The square. Is that right? Am I still online? Can you hear me? Okay. So when we talk about the absolute probability, so this probability A, so we are actually referring to the entire sample space. So if we want to define the value of this probability of A, so we're gonna divide the area of this circle A divided by the entire rectangle, right? Okay. Now, the same thing, if we want to calculate the absolute value for B, so the same thing, we will divide the circle B by the entire rectangle. Is that right? So this is how we calculate the absolute probability. But conditional probability is different because when we talk about the conditional probability, we are not referring to the entire sample space anymore. So for example, if we want to calculate the probability of A given B, so that's the conditional probability. Now, we are not referring the entire sample space anymore. We are not using the the rectangle area. So here, we're gonna use what? We're gonna use, oops, it's actually here. We're gonna use the shaded area divided by B. Does that make sense? So if we want to calculate the conditional probability of A given B, so this probability actually, so if you want to calculate this probability, instead of looking at the entire sample space, this time we only look at the subsample space, B, because we only consider given B, right? So in this calculation, the sample space becomes smaller. And within this B area, we only look at the shaded area. So what's the proportion of the shaded area represented out of this circle B? So this is what we calculate the conditional probability. Is that clear? Okay. Let's go back to here. So this is an example. So suppose we have a sack of marbles. So it's an easy problem because in this sack, we have like 2 blue marbles and 3 red marbles. So if I ask you, what's the chance for you to draw a blue one from this sack? So what is the probability? It's 2 out of 5, right? That's easy. However, if I ask you, what's the probability you draw a blue after taking 1 out? Then there are 2 possibilities, right? So if for the first draw, if you take out a red one, for the next draw, the chance for you to draw a blue should be 1/2, right? But if the first draw was a blue, then the chance for the second draw, the chance of getting a blue one in the second draw should be 1/4. So that means like this is a conditional probability, totally depend on the first draw, the outcome of the first draw. Is that correct? All right. Now the whole sample space is this, but when we calculate the conditional probability, so we do not look at the entire sample space anymore. We look at the subsample space. So this is how we calculate the conditional probability. And if you use this formula here to calculate the conditional probability, yeah, you can easily get this number correct. Any questions here? Okay. All right, so this formula is very important. So when we calculate the conditional probability of A given B, so we always like calculate this ratio. So the probability of A and B, so the joint probability of A and B, which is the shaded area, divided by the circle of B. All right, that's the conditional probability. And if you rearrange this formula, so you can get this. So this is a very important formula for the Bayesian theorem. So in the future, when we talk about Bayesian calculation, so we're going to use this a lot. So yeah, remember this. Any questions? Okay. Now, this is really, really complicated here. So I hope everyone can follow me. So we have 10 minutes. So I hope I can finish this 2 or 3 slides here. All right. So now we have learned the Markov chain, right? And we have learned the conditional probability. Now we're going to use these 2 theorems to help us to calculate the joint distribution, joint probability of any observed genotypes for the 3 loci along a single chromosome. So suppose ABC are arranged in this way on a chromosome. So what's the probability for us to see a specific genotype let's say this. So for example, suppose we are talking about a backcross design. So backcross design, remember, it's very easy. So for each locus, we have two genotypes, which define the state 1 and 2, right? State 1 and 2, state 1 and 2. And we can model this chromosome using a Markov chain here. And if we take out a progeny and do the genotype, if the genotype of a specific progeny is like this, which means A locus is A1, A1. B locus is what? B1, B2, right? And the C locus, C1, C2. All right, so if we get a progeny carrying this genotype at these three loci, so what's the probability of getting this kind of progeny? So this is what we want to do. In this chapter, we want to calculate the joint probability for this genotype. So all right, because of the Markov chain, so we can express this joint probability into this way. Any problems here? Because so the first locus is quite independent. So we put the probability of A here. So because this is the initial state, but when the Markov chain jump from the step 1 to step 2, so if we want to calculate the probability of B, this will depend on the status of the previous step, right? So we use the probability, the conditional probability of B given A here. Let me remove this. This is too messy. Okay. Any questions? So that's why we multiply the probability, the absolute probability of A by the conditional probability of B given A. And once we are dealing with this transition step from B and C, so this is totally independent of the first step A. So we only, when we talk about the probability of C, we only depend on this probability on the step, the previous step, which is B. This is how we understand this formula. Any questions here? So this is an expression for the Markov chain. All right. Now, because the A is the initial step, for A, there's two possible outcomes, right? Either the genotype A or genotype 1 or genotype 2. So they have equal chance, 1/2. That's easy. But when we talk about the probability of a genotype for B, then the situation becomes more complicated, right? All right, so we will generate a matrix. This matrix called the transition probability matrix. So we use T to represent this transition probability matrix, and AB means the transition is between A and B. The transition is between the step A to step B, okay? Okay, so this is a 2x2 matrix. Let's look at, this is a very well designed matrix. It's very useful. You will appreciate the beauty of this matrix in the future, probably next week. Yeah, so here, the first element represented the probability, conditional probability that B carries a genotype 1 given A, the genotype of A is also equal to 1. Can you understand this? So this is the probability that A locus carries a genotype of 1 and B also carries a genotype 1, which means there's no crossing over between these two, right? So what's the probability? The probability should be this. Do you agree? Do you agree? These two things are identical? this element here represented the probability that A and B are both genotype 1. There's no crossing over. So that's why the probability here is 1 minus RAB, right? The same thing here. So the probability, let's look at here. If the first if the locus A and loci A and B are both carrying the genotype 2, so there's no crossing over here, right? So you still see the same probability at this element. So this one is different because this one is the probability that the first locus carry the genotype 1, but the second locus carry the genotype 2, right? Which means there's a crossing over. Then you're going to see the probability this in this position. Okay, the same thing here. So we constructed this beautiful transition probability matrix to facilitate our calculation in the future. So any questions here? Okay, so in the general form, we can say in this transition probability matrix, so we are talking about the two locus, right? The first locus and the second locus. If you see a specific genotype for this two locus, you can use this transition probability matrix to help you to find the right probability based on the recombination frequency, right? So now from here, so this is the probability of the joint probability of A, B, C genotype, and we express this probability, joint probability, using this Markov chain transition probability, right? So this is actually exactly the same with this one, but we use a matrix format here, right? So here we put 1/2 here. What is 1/2? Can anyone tell me?
Probability of A.
Probably, say that again, I'm sorry.
The probability of A being in state 1. Yes.
So because this actually 1/2 is this thing, right? The probability of A. So because for either genotype, the probability is 1/2. That's why we put a 1/2 here. Very good, very good. How about this? The second term. What is the second term? It's actually here, right? And the third term is the transition probability for the next step. Is that clear? All right, now if you have this beautifully defined transition matrix, and if you observe the one genotype like this, you can calculate the probability definitively. So you plug in this 1/2 and using the transition matrix to find the right answer. So from the transition matrix A and B, what's the probability of this? This means like A has the genotype of 2, but B has the genotype of 1, which means there's a crossing over in between, right? The probability should be RAB, right? So that's this thing. And how about this? So this means like for C, genotype is 2, and for B, genotype is also 2. So there's no crossing over between B and C, right? So the probability should be this, right? Is that easy? Yeah, very good, very good. So I really enjoy the matrix, this matrix. The transition matrix is so convenient, so beautiful. And I think I'm gonna stop here for today because there's some solid stuff you need time to digest. Okay, so I'm gonna spend next 10 or 20 minutes to answer your question if you want to discuss now, or if you have some question later, so we can meet in the office hours in the afternoon. So do you have any questions so far?
I have a few.
Yes, go ahead.
So I pretty much have the concepts down.
Mm-hmm.
I do want to go back to Markov chains a little bit.
Sure, give me a second.
Oh.
Markov chain, you mean here?
Yes, sir.
Okay.
Actually, the next slide after that.
Next.
Because as you said, these can get a little bit more complex
Mm-hmm.
when we consider it as A, C, T, and G. So this is just the chart I'm looking at, the flow chart.
Mm-hmm.
This is all based off of the concept of probability distribution is based off of the next state, depends only on the current state.
That's right.
Okay.
Let's see, for example, suppose you are interested in this specific sequence, right? You want to calculate how likely in the nature base, how likely this sequence will occur in this individual. So the starting state is T, right?
Mm-hmm.
And from here. And the next step, so you just think about this sequence is following a Markov chain. The whole thing is the Markov chain. So how many steps in this chain? 1, 2, 3, 4, 5, 6, 7, 8, 9. There are 9 transitions in this chain, right?
Mm-hmm.
So we want to check the 9 steps here. So the first step, the current step, let's say, is T. The second step is T again. So which means like the chain, the first step for the chain will stay in the T state, right? There are 4 states. So you can find that the probability is here, the 0.5, right? Mm-hmm. And if you move forward, the next step out of this 9-step Markov chain, so the next step, the chain transfer from the T state to C state. The probability is another 0.05, right? Something like this. So you just keep going this, and you can use the transition matrix we just introduced to you, but here it's more complicated here because it involves like 4 states and 9 steps rather than the 2 states and the 3 steps, right? So in this example, it's easy because for each step we only have two states. We only involves like 3 steps here, so it's easy. But here, since we are talking about like 9 steps involving 4 states, so the calculation should be more complicated, but still it's doable. So you use the same strategy to calculate the joint probability of this genotype of this sequence. And this will give you how likely this sequence will occur in the nature. If the probability is very high, means, okay, this is probably a random sequence, it's very likely there's no function associated with this sequence. But if the chance of this sequence is very small, which means it is very unlikely for the nature in the nature, it is very unlikely to for a random sequence to evolve into this specific sequence. So which means this sequence is very unique. It is very likely that this sequence is playing a very important biological role. So that will give you some idea, and you can look into this sequence to see if this is true. So something like this. Right?
Interesting. Okay.
Any other questions?
I have a question.
Yes, please.
Can you also show us how the transition matrix will look like for 2-2?
You mean you mean F2?
Yes. No. The states 2, 2. Like if you can go to the last slide.
I see. You mean so
TBC 2, 2. Yeah.
Okay, sure. So in this slide, I only gives you the example for the transition matrix from A and B, right?
Mhm.
So this is actually identical to B and C. They're the same.
Oh,
Yeah, because we're talking about the backcross design. So for each of this loci, locus, for each locus, we only have 2 states, right? 1 and 2. So exactly identical. So you can use the same thing
oh,
for transition matrix B and C. Yes, good question.
okay. Thank you.
Any other
So
questions? Mhm.
so for like the example, like 1/2 TAB TBC, yeah, this is seems like the metric chain, like the TAB matrix multiply matrix TBC. It's not?
So here actually, so actually no, because here we just because for A and B we have a realized value, right? So for example, A equals to 1 and B equals to 2, right? So although this is a matrix, but we only identify a specific individual value to plug in. So this is not actually, maybe I said it in a wrong way.
Okay.
This is not a matrix. Matrix for the next time we're going to talk about matrix calculation. But here we just, based on the realized value for A and B, we identify a single probability value from this matrix. So here,
Okay.
this is only 1, right?
Yeah, we just select the RAV and then
That's
use
right.
the TBC, like this, the
Yes.
data types BC. Okay.
Right. We just select 1 out of these 4 elements from this matrix to use that value for the calculation. So today is a very simple example, but next Tuesday we're going to talk about more
all
complicated
possible,
situation.
like
Yeah,
the all
ways.
possible outcomes.
Yes, yes. General form. So then it's more convenient for us to use the matrix notation. Any other questions? So far, how do you think about the class? You like it or is too hard? Okay, good. Very good.
I'm sure we'll find out on the test.
Sure. Don't worry. So usually, so for this class, let me talk about some history about this class. Yes. So the theorem itself is very complicated. So I try to so it is very hard to teach this class. But usually for the final exam, I do not test the theorem in a very hard way. So I just use a very simple examples to test you. So as long as you understand the principles, it should be okay. It should be okay for you to figure out all the questions. And also, when you try to answer your questions in the final exam, just write down the whole process. Do not just give me the final answer. Just give me everything in the middle. Because even because based on your flow, the logic flow, I can see, I can tell if you understand the theorem topics or not. If you can show me that you understand the whole process, the logic, even you give me a wrong answer, finally, I can still give you the full scores. So because I test you based on your understanding, not based on your calculation. Because for myself, I often make mistakes in the calculation, so I don't blame you. So that's why it is very important for you when you answer my questions in the final. So just show me the steps in the middle, so I can tell whether you can understand the topic, the theorem, or not. Don't worry. So I will not give you extremely hard questions. Yeah. So I teach the hard theorems. I try to teach the difficult theorem in an easy way. So it's not that easy to teach, or it is not that easy for you to learn. But the test should be okay. So don't worry.
About the paper reading, so we need to find the paper by ourselves, or you have some like like the Markov chain papers you can give us and we can read them, or?
No, this is the part of the training. I think for a future scientist or outstanding researcher like yourself. So if you have a couple of keywords, you can easily find the literature by yourself.
Okay.
Yeah, yeah. I think it's not that hard. So because I have taught this class for a couple of times, and each time I think students are doing great in terms of the term papers. You can easily find tons of papers. By using some of the keywords. The only problem is like probably you need to spend some time to narrow down to find the best paper you should use for the presentation.
Just as a clarification, Professor,
Mm-hmm.
well, it isn't always advised. There will be some topics that will be covered closer to the term papers, so we might have to do a little bit of reading ahead if we wish to cover one of those topics.
Yes. So I don't think I don't know if I understand the question. So you mean, so suppose you want to talk about some topics which has not been covered so far, because we're learning the chapter 4 right now. So do you need to read the textbook ahead of time? Is this your question?
Yes, it's possible that we might need to read the textbook ahead of time a little bit.
I don't think so, because I know all of you are very busy, so you have other classes to take, and you are also probably teaching as a TA, so you do not have time to read the textbook ahead of time that far. But yeah, it is always helpful if you can read the textbook before my lecture. But for the term paper, I think yeah, you can wait till like probably like maybe week 7 or week 8. So at that time, probably you have some idea which topic you are most interested in, and then you can start looking into the literature to find some papers. I don't think it will eat up too much of your time, so don't worry about that. You can wait till like maybe week 7, week 8 to start doing that.
Got it. Thank you.
Sure. Any other questions? All right, very good. Okay, so if you do not have questions, we're gonna dismiss the class for now. So in case you have questions later, so you can either join the office hours in the afternoon, or you can shoot me an email. So I try to answer your questions by email. All right. Otherwise, see you next time. Enjoy the weekend. Bye-bye.
Bye.
Bye.